Hello, good evening students. Today we are going to discuss about a complete information about the prokaryotic cell. Already the best example for prokaryotic cell is a bacteria cell. So today we are going to discuss a complete information how the cell structure, what kind of cell organelles are present in the bacteria cell, and how the cell wall and remaining membranes they are arranged in the bacteria cells. So everything a complete information today I am going to discuss. So this is the third video of this chapter. So this is a neat biology chapter. So I will give full information about the neat subject, both botany and zoology. Okay students, now let us go to the topic bacteria cell structure. Bacteria cell structure. So in the previous video just I will give an information. Bacteria cell is enclosed by three membranes. So what are the membranes? See the diagram. I will draw the bacteria cell diagram. So this is about a bacteria cell, and this bacteria cell is enclosed by the three membranes. So one is outer membrane, and second one is middle membrane, and also the innermost membrane also present like this. So commonly the bacteria cell is enclosed by the three membranes. So first, what is the innermost membrane of the bacteria? See, the innermost membrane of the bacteria is called plasma membrane. Plasma membrane. So outside of the plasma membrane, it consists of one more membrane here. This membrane is called cell wall. Then the outermost layer of the bacteria cell is called glycocalyx. Glycocalyx. So like these three membranes are enclosed by the bacteria cell. In this content, now I discuss only the bacteria cell wall. So you have to remember one thing. Three membranes together we are called as one name. Three membranes, glycocalyx, cell wall and plasma membrane. Together we are called as a cell envelope. So what is cell envelope? The plasma membrane plus cell wall plus glycocalyx, together we are called as a cell envelope. Then these membranes are arranged one above another, like this plasma membrane above the cell wall and above the glycocalyx. See, especially the cell wall is made up of peptidoglycan. Peptidoglycan. This is one type of sugar. Remember that important, peptidoglycan. Then how these membranes are arranged means, if we take these membranes in separate diagram, I will draw some portion of the membranes here. See, this is the outermost membrane, and second one is the middle membrane. See, this is called middle membrane, and then innermost membrane like this. I will draw here only the membranes. So they are arranged one above another. See, these membranes Already we know this is a plasma membrane and this is called cell wall and this is called glycocalyx. Why I will draw in separately means bacterias are also, all bacterias are classified into two types based on their envelope. So what are the types of that bacteria? See, the bacteria are majorly classified into two types. One is gram positive bacteria, another one is gram negative bacteria. So how to determine, how to find out which is positive bacteria and which is gram negative bacteria? We are going to conduct one experiment. The experiment is means gram stain. So what is gram stain? Gram stain means giving stain to the bacteria cell. Stain means color. So in this method we will follow the 3 steps in this method, in the gram staining method. So what are the methods? Now I will discuss. See, we are going to discuss about the types of bacteria. Types of bacteria based on the envelope. Based on the envelope. So already you know 3 membranes together we are called as a cell envelope. Okay, based on the envelope, see first there are majorly 2 types of bacteria. First one is gram positive bacteria. Gram positive bacteria, and second type of bacteria is gram negative bacteria. So how to find out which is gram positive and which is gram negative? With the help of one process, gram stain or gram staining. So gram staining means addition of colors or chemicals to the bacteria cell. See, for suppose I will discuss about the gram positive bacteria. How to determine? See, the gram positive bacteria, we will take 2 membranes, no need to outermost membrane. For that reason, I will draw here only 2 membranes. One is outer membrane and second one is innermost membrane, like this. 2 membranes alone. No need glycocalyx for gram staining. For that I will draw only the 2 membranes. So the innermost membrane is called plasma membrane and the outermost membrane is called cell wall. So in this experiment we will take some chemicals and dyes. Dyes means colors. So in the first step, so we are following majorly 3 steps. So what about the first step? In the first step we are taking the crystal violet solution. Crystal violet plus iodine solution. The combination of crystal violet and iodine solution is added into the bacteria cell. Then this crystal violet along with the iodine solution which is observed by the cell wall. The cell wall observes that colors or chemicals, and they form a blue color in between the cell wall and plasma membrane. In between the cell wall and plasma membrane, it gives blue color. It forms a blue color. So in the first step, in between the cell wall and plasma membrane, the blue color will form. So blue color. Then going to the second step. In the second step, we add the alcohol or acetone. Alcohol or acetone, anyone, to the cell wall. Then here the alcohol or acetone, which is get mixed with the blue color, again it forms a blue color only. In the second step also it forms the blue color. So now coming to the third step. What is the third step? In the third step, we add saffron into the cell wall. So actually saffron is a pink color dye. Pink color dye. So then it also get mixed with this and it also forms a blue color only. It also forms a blue color. See, during the process of gram staining, we will follow the three steps. In the first step, crystal violet plus iodine solution given to the cell wall. The cell wall absorbs that crystal violet and iodine and it forms blue color in between the cell wall and plasma membrane. In the second step, we add the alcohol and acetone. This also gives blue color. Then finally we add a pink color saffron. It also forms blue color. So when we are applying the gram staining method into the bacteria, it forms always blue color. So this blue color indicates this is a gram positive bacteria. This is a gram positive bacteria. Then going to the gram negative bacteria. See, here also I will draw the two membranes. See, the outer membrane is called cell wall and inner membrane is called plasma membrane. See, this is plasma membrane, this is cell wall. So here one specialization is there. In some bacteria, the cell wall is not a single membrane. Some portion of the cell wall is also having some other substances. The cell wall, some portion of the cell wall, it is made up of some other material. Commonly, see here, this is the some portion of the cell wall. This is also cell wall. These two membranes together we are called as a cell wall. Commonly, cell wall is made up of peptidoglycan. Already we discussed peptidoglycan. And the above the membrane is made up of lipoproteins. So these lipoproteins are it does not allows the some substances into inside of the cell wall. So the same steps we are following the gram negative bacteria. See here, the same step. First, the crystal violet plus iodine solution given to the cell wall here. Then it also reaches into the middle of the plasma membrane and cell wall, and it forms blue color here also. In the first step, it also form blue color. See the same step. Then going to the step 2, alcohol or acetone is given to this cell wall, alcohol or acetone. So the alcohol or acetone which is react with this iodine plus crystal violet solution, then it is not accepted into this, then this blue color dye, it will be come out from the cell wall into outside, then this it will be converted into the blue color is converted into colorless here, so in the second step, so the blue color is converted into colorless, which is comes into outside of the cell wall. So in the second step, colorless, the substance is converted into colorless. So then going to the third step, the same step, the saffron and pink dye. So here there is no blue color. Imagine that there is no blue color here. Why there is no blue color? It will be eliminated from the cell wall into outside. So there is no blue color. Then we add saffron and pink dye into this. Then what will happen? Here the pink color will be appears in between the cell wall and plasma membrane. So in the third step, the pink color will appear. So when we apply the gram staining method into the bacteria, finally it forms a pink color. It shows the pink color. This pink color indicates it is a gram negative bacteria. So this is the conclusion of this, which is gram positive and which is gram negative bacteria. Then we are going to the some of other information of the bacteria. Okay. So again I will draw the what are the structures are present in inside of the bacteria cell. See, already you know very well, the outermost membrane is the bacteria. So this is the bacteria cell. What is the name of outermost membrane? Glycocalyx. Then going to the second membrane, the second membrane of the bacteria, which is inside of the glycocalyx. This membrane is called as a cell wall. Then coming to the innermost layer, the innermost layer of the bacteria, this membrane is called as a plasma membrane. So these are the three membranes. And what are the innermost organs are present in inside of the bacteria cell or inside of the plasma membrane? See, here some important chromosome is present here, like this. So This is a chromosome. Bacteria cell chromosome. The bacteria cell chromosome, they are not having the histone protein. Without histone protein, commonly the histone protein present in the eukaryotic organism chromosomes only, but in prokaryotic organism there is no histone protein. Remember that only the histone protein present in eukaryotes only. Okay, some portion of the chromosome like this. Already the chromosomes are made up of DNA. Some portion of the chromosome, it is modified into a long thread-like structure like this. This long thread-like structure is called mRNA. This is called what? mRNA. So in this mRNA, some ribosomes are attached to the mRNA like this. Ribosomes are commonly two subunits. Already you know very well. Like this, some of the ribosomes which are attached on the mRNA. This kind of ribosomes we are called as a polysomes. Poly means many. Polysomes. So some of the other ribosomes, they are present in the cytoplasm of the cell like a individual, like this individual. These are not group, free ribosomes. So these are free ribosomes. Free ribosomes. And a group of ribosomes which are attached on the RNA is called, mRNA is called polysomes. And also some portion of the chromosome, like a portion of the chromosome is called DNA. Remember, very important. The chromosomes are made up of DNA and proteins. So some portion of the other portion of the DNA, it also, this is called DNA. It is modified into circular DNA here. This circular DNA is called plasmid. The circular DNA is called plasmid. So the plasmid is not necessary for survival of the bacteria cell. Remember that. So it is not necessary for survival of bacteria cell. But what is the role of this DNA or plasmid means it resistant to the tetracycline, to resistant, or it resistance to tetracycline. Remember, so it resistant to tetracycline. So these are the structures which are present in the inside of the plasma membrane in the prokaryotic cell or bacteria cell. Now some of the other structures are also present in this bacteria. So what are the other structures? See, I will rub some parts for explanation purpose. If you want now, you take a screenshot and I will go to explain the remaining parts of the bacteria cell. Okay, see, what are the some other parts present in the bacteria cell? See, the bacteria cell having a long flagella here like this. See, this is the flagella. So this structure is called flagella. So look like a tail-like structure which is helpful for movement or locomotion. Movement or locomotion. Locomotion of bacteria cell. So it also consists of flagella, and the flagella is also consists of some important parts like this, the innermost box like structure. This is called as a basal body, and this tail like structure is called filament. So these are the parts which are present in the bacterial flagella, and also it also consists of some other tiny hair-like structures like this. Around the bacteria, it also having some tiny hair-like projections. These tiny hair-like projections are called cilia. So the cilia is also helpful for movement of bacteria. Movement of bacteria. So what are which organs are helpful for movement of bacteria means cilia and flagella. Remember that, cilia and flagella. And also some finger-like projections are also present here. Some of the finger-like projections like this. Around the bacteria, some of the finger-like projections also present. So these finger-like projections are called fimbriae. These fimbriae is helpful for to hold the other organisms. So when the bacteria hold any other organisms for nutrition purpose, it hold the other organisms with the help of these finger-like projections. These finger-like projections are called fimbriae. So what purpose means to hold the other organisms. Other organisms. So this is the function of the fimbriae. Then one more important organ is also present here. Some of the, see, look like this, like this. Some of these structures which is present among the glycocalus or outermost membrane, these structures are called villi. So what is the function of villi means, if the bacteria transports the genetic material, one bacteria, it transports the genetic material from one bacteria to another bacteria through this villi. So what is the purpose? Transfer of genetic material. Transfer of genetic material from one bacteria to other. So that is the function of the villi. And also here the bacteria cell having the double stranded plasmid Remember, the plasmid is not necessary for the cell survival, bacteria cell survival. But what is the use of this plasmid means, commonly, for suppose this is one bacteria cell which consists of the plasmid, this is called plasmid, and some of the other bacteria they lack the plasmid. Some of the bacteria they lack plasmid. Then this plasmid is given to the other bacteria. And also in some bacteria they have some specialized gene here. Some genes are present here. This gene is work as a antibiotic. Some other bacteria they lack this gene, and which bacteria having this gene is transferred to the other bacteria. See, this gene is transferred to the other bacteria. So that is the use of plasmid. So this is the information about the bacteria cell structure. So then in the next video we are going to discuss about the eukaryotic cell. So dear students, మీకు ఈ వీడియో నచ్చినట్లయితే ఖచ్చితంగా లైక్ చేయండి, సబ్స్క్రైబ్ చేయండి. ఈ వీడియోని మన వీడియోని అందరికీ మీ ఫ్రెండ్స్ అందరికీ షేర్ చేయండి. మీరు ఇచ్చే సపోర్ట్ మాకు చాలా హెల్ప్ఫుల్గా ఉంటుంది. నేను ఖచ్చితంగా నీట్ బయాలజీ సంబంధించి బాటనీ, జువాలజీ, ఈచ్ అండ్ ఎవ్రీ టాపిక్ ప్రతిదీ కూడా వీడియో చేయడానికి నేను ట్రై చేస్తాను. ప్లీజ్ గివ్ మీ యువర్ సపోర్ట్. Okay, thank you. Now we are going to meet in the next video. In the next video we will discuss in a structure of eukaryotic cells. Okay, thank you.